Protein synthesis. So now we're going to talk about how our cells make proteins. And if you remember, proteins are one of those macromolecules that your cells, they just cannot live without. Protein synthesis is the process of reading the instructions inside of your DNA to make a polypeptide. If you remember from macromolecules, amino acids, right here, they linked together to form a polypeptide chain. So the amino acid is the protein monomer, and the polypeptide chain is the polymer. When we create this link, that is our primary structure. When the amino acids are in the correct sequence, we get a primary structure chain linked together. And then the protein will start to twist on itself, forming the alpha helix. There's also a beta sheet, looks slightly different than this helix. And once it starts to form this helix, it'll form a tertiary structure where it'll start to fold in onto itself. And then if we get a couple of tertiary structures together, we're going to form this quaternary structure where the proteins are all bonded together. So two or more of these polypeptide chains bound together is a quaternary structure. And a polypeptide chain will fold onto itself. And remember that the form or the shape of the protein is function. Its shape is going to determine what it does. For this lesson, for protein synthesis, we're going to be talking about this guy right here, the polypeptide chain amino acid sequence, and what goes on to make that protein. Central dogma is the process in which we go from DNA to protein. If you remember, DNA is inside the nucleus. It's a very bulky molecule. It can't leave the nucleus. It's too big. But the instructions in order to make everything are inside of that nucleus. This blue line is my nucleus, and inside of it is the DNA. Here we have the cell, and inside of cells are our ribosomes. So in order for this to occur, we're going to have a two-step process, transcription and then translation. Transcription is going to be the instructions that code for the RNA molecule, specifically we'll be talking about mRNA, that are going to leave the nucleus. And then once that RNA is inside of the cell, finds a ribosome, we're going to translate that into a protein. And if you remember, the ribosomes, they are on the rough endoplasmic reticulum, and they're also floating in the cytoplasm. So there's two different locations at which that mRNA can be translated into a protein. So how I remember this is that the first step, if you want to copy something, you are going to transcribe it. You're going to go through transcription. If you want a copy of all of your grades, then you go to the front office and you ask them for a transcript. When you apply to colleges, secondary education, they want a copy of everything in high school, so they are going to ask for your transcripts. So a transcript is the copy, and then they take that copy and they translate it into what you're going to need for your college or secondary post-high school education. Everybody's going to translate that transcript a little differently depending upon your acceptance or what you're going to need to fill in gaps. But that's just how I remember the process of central dogma genetics. So if you look right here, here's our DNA molecule. It's a double helix. It's way too large, so we're going to get a copy of it, transcription, to form RNA, which will be a single strand right here that is going to leave the nucleus. And then inside inside of the cell, this mRNA is going to look for a ribosome. And when it finds a ribosome, either on the rough endoplasmic reticulum or floating around in the cytoplasm, the process it's going to go through is translation. So your copy is going to be translated into a protein.